Welcome to today's deep dive into apolipoproteins, the crucial proteins that manage our body's lipid transport system. Whether you're preparing for your ASCP certification, studying biochemistry, or just wanting to understand lipid metabolism better, this video will break down everything you need to know about these fascinating proteins. Let's get started. Before we dive into specific types, let's understand what apolipoproteins are and why they matter. Apolipoproteins are the protein components of lipoproteins, serving three main functions. First, they activate enzymes involved in lipoprotein metabolism. Second, they maintain the structural integrity of lipoprotein complexes. And third, they facilitate lipoprotein uptake by interacting with cell surface receptors. Think of them as both the structural framework and the traffic controllers of lipid transport in our bloodstream. Now, let's talk about distribution. Apolipoproteins are present in all lipoproteins, with a notable exception, low-density lipoprotein, or LDL, which mainly contains Apolipoprotein B100. In high-density lipoprotein, or HDL, Apolipoprotein A1 is predominant, while other apolipoproteins, like C1, C2, C3, and E, appear in various proportions across lipoproteins. It's also interesting to note that lipoproteins can weakly bind additional plasma proteins. However, the exact roles these proteins play in metabolism remain somewhat unclear, indicating that there's still much to learn in this area. Let's zoom in on some specific apolipoproteins, starting with Apolipoprotein A. Apolipoprotein A contains an amphipathic helix. This means it has a hydrophobic side that interacts with the lipid core of lipoproteins, while its polar or charged side faces outward. This unique structure allows apolipoproteins to exchange between lipoproteins during metabolism, showcasing their dynamic nature. Apolipoprotein A1 is particularly important. It constitutes around 90% of the protein content in HDL, working alongside Apolipoprotein A2. Its functions are impressive. It serves as a structural component of HDL. It acts as a cofactor for LCAT, an enzyme that esterifies free cholesterol. It functions as a ligand for ABCA1, a sterol efflux protein. Moreover, ApoA1 can appear in 1 to 5 copies per HDL particle, significantly affecting particle size and structure. Now, let's talk about Apolipoprotein A2. In HDL, the ratio of ApoA2 to ApoA1 is roughly 1 to 3. It has some intriguing potential roles, such as inhibiting LCAT and activating hepatic triglyceride lipase. This means it plays a part in delaying the breakdown of large triglyceride-rich lipoproteins. Moving on to Apolipoprotein A4. Synthesized in the intestine, it's secreted as part of chylomicrons. ApoA4 helps facilitate the release of ApoC2 from HDL or VLDL and enhances intestinal lipid absorption while also influencing satiety through hypothalamic pathways. Lastly, we have Apolipoprotein A5. Though it's found in low abundance, its functions are critical as it modulates triglyceride concentrations and facilitates interactions between triglyceride-rich lipoproteins and lipoprotein lipase. Next, we're going to explore Apolipoprotein B. Apolipoprotein B, or ApoB as it's commonly known, is pivotal in our body's lipid metabolism. It comes in two primary forms, ApoB100 and ApoB48. Each plays a unique role in how our body processes and utilizes lipids. Let's begin with ApoB100. This is the full-length version, comprising over 4,500 amino acids, and it's primarily produced in the liver. Its journey starts as part of very low-density lipoproteins or VLDL. As VLDLs shed their triglycerides, they transition into low-density lipoproteins, or LDL, commonly known as bad cholesterol. ApoB100 is integral in this process, serving as the main apolipoprotein in LDL. Why is this important? Well, it acts as a surrogate marker for LDL particle concentration, often abbreviated as LDLP. This means that when you hear your doctor discussing cholesterol tests, ApoB 100 is one of the markers they're considering. Moreover, it facilitates the receptor-mediated internalization of LDL in both hepatic and peripheral tissues. It essentially ensures that LDL reaches where it needs to go within your body's cells. Structurally, ApoB 100 is a marvel. It contains amphipathic helices and beta sheets, which create an incredibly high lipid-binding affinity. This feature is crucial because it prevents the transfer of ApoB 100 between different lipoprotein particles during metabolic transformations. Now, onto ApoB 48, a shorter sibling of ApoB 100, with 2,152 amino acids. This version is synthesized in the intestines. Its production is fascinating. It involves RNA editing that introduces a stop codon at residue 2153 of ApoB 100. ApoB 48 is the primary apolipoprotein of chylomicrons, essential particles that transport dietary lipids from the intestines to other parts of the body. Without ApoB 48, chylomicron secretion would be severely impaired. While ApoB 100 and ApoB 48 have different origins and roles, both are crucial for the secretion of lipoproteins, ApoB 100 for VLDL and ApoB 48 for chylomicrons. ApoB 100 also interacts with LDL receptors, a vital process for cholesterol clearance in the body. However, it's worth noting that certain proteins, such as ApoC3 and ApoA, can block this binding. This blockage impairs the clearance of particular particles like lipoprotein A, commonly referred to as Lp(a). The next stop will be Apolipoprotein C family. The Apolipoprotein C family comprises 4 primary members, ApoC1, C2, C3, and C4. Among them, ApoC1, C2, and C3 are present in significant amounts in human serum, whereas ApoC4 isn't. These proteins are primarily synthesized in the liver, with some production in the intestine. Once synthesized, they are distributed throughout various parts of the body. Notably, ApoC1 and C2 are present on all lipoproteins except LDL, while ApoC3 is found on LDL as well. Let's start by examining ApoC1, the smallest of the C apolipoproteins. Comprising just 57 amino acids, its role is crucial. It activates LCAT, an enzyme essential for cholesterol metabolism, while inhibiting several key enzymes, including lipoprotein lipase and CETP. Its significance is most prominent in the inhibitory activity within HDL particles. Next, we have ApoC2, consisting of 78 amino acids. Its function is pivotal for triglyceride metabolism, as it activates lipoprotein lipase. This activation is essential for breaking down triglycerides into fatty acids. ApoC3, on the other hand, is a bit more complex. It's a glycoprotein made up of 79 amino acids and exists in 3 different isoforms due to variations in sialic acid content. These isoforms play significant roles in regulating triglyceride levels and VLDL metabolism. Interestingly, recent insights reveal that ApoC3 doesn't directly inhibit lipolysis by lipoprotein lipase, as previously thought. ApoC3 is also implicated in inflammatory pathways, adding another layer to its complex biological profile. Genetic studies have shown that loss-of-function mutations in the ApoC gene are linked with favorable lipid profiles and lower coronary artery disease risk. Clinically, ApoC3 has been highlighted in major studies like CARE, the Physician's Health Study, and the Women's Health Study for its role in atherogenesis. Due to its significant impact, targeting ApoC3 could provide promising therapeutic avenues for managing hypertriglyceridemia and coronary artery disease. Antisense oligonucleotide therapies targeting ApoC3 synthesis show potential and are currently undergoing clinical trials. Lastly, we have Apolipoprotein E, or ApoE. Apolipoprotein E is a 34 kilodalton plasma glycoprotein composed of 299 amino acids. Primarily synthesized in the liver, ApoE is also made in the brain, macrophages, and many other tissues. This breadth of synthesis allows ApoE to be a major player on the scene of lipoprotein regulation within our bodies. ApoE is omnipresent on all lipoproteins, although its presence on low-density lipoproteins, or LDL, is minimal. Its key function is to facilitate the uptake of lipoproteins in the liver by interacting with receptors, such as the chylomicron remnant receptor and the LDL receptor-related protein. There are 3 common isoforms of ApoE, E2, E3, and E4, each differing by specific amino acids at positions 112 and 158. These differences lead to variations in binding affinities and influence lipid levels in the body. The clinical implications of ApoE are significant, particularly concerning cholesterol synthesis and diet. For instance, the ApoE4 isoform is linked with increased cholesterol absorption, historically advantageous on calorie-restricted diets, but now associated with a higher risk of coronary heart disease. Interestingly, ApoE4 carriers often show reduced responsiveness to statins. However, meta-analyses suggest there's limited benefit in using apogenetic testing to guide statin therapy. The ApoE4 allele story doesn't end at cholesterol, it extends deeply into our neurological health. It's strongly correlated with a higher risk of Alzheimer's and other neurological disorders, with current research suggesting that risk is potentially tied to ApoE concentration. Thank you for joining me, and be sure to stay curious as we learn. Until next time. Bye.